Hello and welcome to Unit 1.1, Monomers and Polymers. I will begin by quickly outlining what Unit 1 is all about. All life on Earth shares a common chemistry. This provides indirect evidence for evolution. Despite their great variety, the cells of all living organisms contain only a few groups of carbon-based compounds that interact in similar ways. Carbohydrates are commonly used by cells as respiratory substrates. They also form structural components in plasma membranes and cell walls. Lipids have many uses, including the bilayer of plasma membranes, certain hormones and as respiratory substrates. Proteins form many cell structures. They are also important as enzymes, chemical messengers and components of the blood. Nucleic acids carry the genetic code for the production of proteins. The genetic code is common to viruses and to all living organisms, providing evidence for evolution. The most common component of cells is water, hence our search for life elsewhere in the universe involves a search for liquid water. Now that I have outlined what Unit 1 is about, welcome to Unit 1.1, Monomers and Polymers. What are monomers? Monomers are small, identical or similar molecules, that can be joined together to make larger molecules, called polymers. Polymers are large molecules which are formed by joining many identical or very similar monomers together. Nucleic acids, amino acids, alpha and beta glucose, fructose, fatty acids and glycerol are all examples of monomers. The three main polymers you need to know are, number 1, carbohydrates. Monomers are joined by glycosidic bonds. Number 2, proteins. Monomers are joined by peptide bonds. And number 3, lipids. Monomers are joined by ester bonds. The formation of polymers occurs through condensation reactions. A condensation reaction joins two molecules together with the formation of a chemical bond and involves the elimination of a molecule of water. Polymers are then broken down through hydrolysis. A hydrolysis reaction breaks a chemical bond between two molecules and involves the use of a water molecule. Condensation reactions produce water. Hydrolysis reactions use water. Now, let's look at examples of monomers in more detail. One example is a nucleotide. A nucleotide is made up of three components: a nitrogenous base, a pentose sugar and one or more phosphate groups. Two types of pentose sugar are found in nucleotides: deoxyribose, found in DNA, and ribose, found in RNA. Deoxyribose is similar in structure to ribose, but it has an H instead of an O at the two positions. The diagram on screen now shows the structure of nucleotides. When nucleotides join together they can form RNA or DNA, depending on whether the nucleotides have ribose or deoxyribose. They form phosphodiester bonds. Another example of a monomer is glucose. Glucose is a hexose sugar, a monosaccharide, which has 6 carbon atoms in each molecule. The two types of glucose are alpha and beta. They are isomers, which means they have the same molecular formula, but the atoms are connected in a different way. In your exam, you need to know the structure of both types of glucose. This is actually easy because there's only one difference between them, as the diagram on screen shows. The oxygen and OH group on carbon 1 on the right side are switched. On alpha glucose, the hydrogen is on top. On beta glucose, the hydrogen is on the bottom. This affects the way that glucose polymers form. Starch and glycogen are made when alpha glucose join together and form many 1 to 4 glycosidic bonds. They have 1 to 6 branches. Cellulose is made from beta glucose. We will look at proteins and lipids in more detail later on. To recap, monomers are the smaller molecules which make up polymers. Polymers are formed through condensation reactions which produce water. They are broken down through hydrolysis which uses up water. Carbohydrates are made from monomers joined together with glycosidic bonds. Proteins are made from amino acids joined together with peptide bonds. Lipids are made from fatty acid and glycerol, joined together by ester bonds. There are two isomers of glucose, alpha and beta. On alpha, the hydrogen on carbon 1 is on top. On beta glucose, the hydroxyl group is on top. Thank you for watching Phreology Revision.